This is the second part of our series on vaccine immunology. In this chapter, we will learn about antibodies, their structure, function, and amazing versatility in helping to protect our bodies. Antibodies are produced by specialized lymphocytes, known as B cells. Antibodies present on the B cell surface function as B cell receptors. Antibodies can also be secreted into the blood, lymphatics, and mucosal surfaces as soluble immunoglobulins. Antibodies are immune system-related proteins called immunoglobulins. Each antibody consists of 4 polypeptides, 2 heavy chains and 2 light chains, joined to form a Y-shaped molecule. This structure allows antibody molecules to carry out their dual functions, antigen binding, and biological activity mediation. The variable section contains the hypervariable regions, with a high ratio of different amino acid combinations. This highly variable structure equipped the antibody with the ability to bind with a large variety of antigens. The constant region determines the major antibody classes, and determines the mechanism of action. Antibodies are divided into 5 major classes: IgG, IgM, IgA, IgD, and IgE, based on their constant region structure and immune function. Different classes of antibodies or immunoglobulins have different characteristics. IgG is the most abundant class of antibody in the human body. It exists as a monomer, and is found ubiquitously in the blood and lymphatics. IgM consists of 5 immunoglobulins, combined to give it its classical pentamer structure. It has the highest molecular weight, and is also widely distributed in blood and lymphatics. IgA is dimer, and is found on mucosal surfaces, secretions, and blood. IgD and IgE are monomers which are found in small amounts compared to the other antibodies. Each immunoglobulin class has different functions. IgM is the first antibody to appear during an acute immune response, and serves to fixate complement, and forms part of the activated B cell receptor. IgG antibodies are formed at a later phase after appearance of the IgM antibodies. IgG is the main antibody in the body, and it serves to neutralize toxin, and facilitate opsonization. IgA are secreted into mucus, tears, and saliva, and forms a first line of defense toward invading pathogen. IgE is activated in allergic reactions, and also has an important role in antiparasitic activity. IgD forms part of the B cell receptor, and acts to facilitate B cell activation in an immune response. Our body's antibodies are immensely diverse, and versatile. Due to its inherent structure, it has an almost unlimited potential to recognize a huge repertoire of antigens. The ability of our antibodies to cross-react with a variety of antigens also means that it can potentially react to antigens which the body has never encountered before. Antibodies exert their function in a variety of ways to inactivate or destroy foreign substances. First, antibodies can bind to and inactivate foreign substances such as pathogens and toxins, a process called neutralization. Secondly, antibodies can activate the complement system to destroy bacterial cells by lysis. Antibodies can also facilitate phagocytosis of foreign substances by phagocytic cells, a process called opsonization. To understand more about human antibodies, we recommend that you take a look at this video from nature.com, which illustrates the complex diversity of our human antibodies.
People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites. And for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins. And billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes. But they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of one V, one D, and one J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.